Assalamualaikum and good evening to the Dr. Andrew. Today I would like to present about PCR. Okay. So what is PCR? PCR is stand for Polymerase chain reaction. The DNA copies produced through PCR amplification can be used in large number of medical and also forensic application. But why PCR is very important? So the first one, PCR is very important to the medical application. Why is medical application is important? Because PCR was first used to analyze the presence of genetic disease mutation by do the genetic testing. For the forensic application, PCR is used for determine the genetic fingerprinting to incriminate or rule out suspect in a crime investigation. While for the infectious disease and detection and identification, PCR is used to detect the HIV and other diseases such as ear infection, tuberculosis and Lyme disease. And lastly, it can be application in molecular biology research, which is DNA sequencing, the DNA cloning and gene expression. PCR can be used to produce huge number of pure DNA sample from a limited source. So gene, it can be also amplified using this copying process by using PCR. The process rather like DNA replication, in that each strand of duplex double-stranded DNA is copied, the duplex split apart and the copying repeated over and over again. Next, what is the factor that occurring of the genetic variation? So the first one is mutation, the second one is random mating, the third one is random fertilization, and the last one is recombination between homologous chromosome during meiosis, which is reshuffle allele within an organism offspring. Okay, next we move to the mutations. So what I understand about the mutation change the protein function. So in this part, the mutation change of their protein function is called by missense mutation. So it is a mistake in the DNA which result in the wrong amino acid being incorporated into a protein. Because of change, that single DNA sequence change result in a different amino acid codon which the ribosome recognize. Change in amino acid can be very important in the function of protein. Sometimes missense mutation cause amino acid to be incorporated, which make the protein more effective in doing its job. So, more frequently, it causes the protein to be less effective in doing its job. But this is really degrees of evolution when missense mutation happen, and therefore small change. Frequently, small change in protein happen, and it happens to be that improve the function of protein. So, this page, I insert the picture, which is can see the missense mutation at the sequence of the DNA. We move to the why mutation important to the plant. Firstly, the usefulness of mutation breeding in crop improvement was demonstrated firstly in Sweden, which embarked upon practical plant breeding of agricultural plants by means of X-ray and also ultraviolet induced mutation. So the Swedish plant breeders found many chlorophyll mutations in the belly by the use of X-ray. They discovered some mutant characterized by denser head, late maturity and very stiff taller straw. These mutants were found to yield higher and produce more straw than the maternal variety. So the first one is important is improvement of traditional traits like yield, to their resistance toward disease and also pest. The second one, mutation is very important because it can create a new species of plant. And next, also it can be crop improvement and solving the issue of related to the world food security. And then lastly, the development of dwarf wheat and also rice variety that lead to the green revolution is a classic example in the mutation breeding. through the successful exploitation of the mutant of gene. So that's all for me. Thank you.